and that's fine, but I'll go ahead and get started. So it's my pleasure today to introduce our speaker, Ashley Blawas. Did I say that right? Yeah. Okay. So I first met Ashley virtually a few years ago when she asked me to be on her PhD committee. It was a great committee to be on because she did an outstanding thesis on the physiology of diving, or diving physiology, which you all, or most of you know, that I love. But she went from the scale of the cell all the way up to the whole animal, so broad range. She used tools from molecular biology, comparative physiology, and another thing I love, biologging. She completed her PhD from Duke in 2023, and then shortly after, she moved out to California and started a postdoc in the Goldbogen lab at Hopkins. So then we actually finally got to meet in person after some virtual meetings. At Stanford, she studies the physiology of baleen whales off of our coast using biologging tags, and then she has been developing the capacity for physiologging by engineering novel designs for EKG-equipped tags, so trying to measure heart rate in baleen whales. Her ongoing research includes understanding the scaling of physiological rates in cetaceans and the molecular drivers of extreme cardiac function in the diving baleen whales. So, as you can tell, she works at the intersection of marine mammal science, engineering, and ecological physiology to investigate the physiological traits that underpin the extreme metabolic function of cetaceans. To date, her work has led to new insights that inform our understanding of basic physiological principles, as well as translational medicine and conservation. She has published an impressive 13 papers, considering she's only had her PhD for about an hour and a half, a year and a half. That would be really impressive.
That would be really cool.
And one book chapter. And then in addition to being an excellent researcher, she also values teaching and mentoring. So she was a TA at Duke, but she has also co-taught a course in biologging. And those of you in the biologging course are going to get to have her as a guest lecturer while I'm gone. And she's also mentored students at Salinas High School and through the CSU MB REU program. So with that, I'm excited to hear what she has to say. So thank you.
Thank you, Gita. Hi, everyone. Thanks for being here. As Gita said, my name is Ashley, and I'm a postdoc in Jeremy Goldbogen's lab just down the road at Hopkins Marine Station. And today, I'm going to be presenting work that was part of the latter stages of my PhD, as well as work that's ongoing in my position as a postdoc on methods to sense or measure physiology in whales. And I want to point out before I get too deep into the talk that I'm sort of using the royal whales in the title here, and that really the talk today is going to cover cetaceans broadly, cetaceans being the taxa that includes whales, dolphins, and porpoises. And I want to thank Gita for the invitation to be here and for the lovely intro. I wrote this in my notes, but as Gita said, she was on my dissertation committee at Duke, and we met virtually many times back then, so it's really cool to be here in person at Moss and give this talk. Before I get into the meat and potatoes of the seminar, I want to take a second to acknowledge the many, many collaborators that have contributed to the work I'm going to present, all the way from ideation to fieldwork and data collection and funding. As I'm sure you all are very aware in this room, field-based research takes large teams of highly skilled individuals across a range of skill sets, many that I do not possess myself. And so these people that are listed here, as well as many others that I probably forgot about, are really truly mission critical to the work that I've done and will continue to do, and I want to make sure that they're recognized for that contribution. So today I'm going to be talking about cetacean physiology, and particularly the pitch that I'm hoping I can successfully make is that there's a lot we can learn from investigating ways to sense the physiological function of cetaceans because of the extreme nature of the taxon as a whole. And I want to start off my talk focused on the animal on this slide, which I'm sure you all know is a blue whale, because this species demonstrates extreme function in a way that is perhaps most evident to us as visual creatures, and that is their size. So blue whales are 20 to 25 meters long. I meant to walk across this room and measure how long it is. I don't think it's 25 meters. So they're really big animals. They're the largest to have ever existed on planet Earth. And in our lab, the Goldbogen lab, we study these species amongst other large whales to understand, and the analogy that I'm going to use today is the recipes that have resulted in the evolution of the largest animals on the planet. And we do that through a variety of different lenses based on the specialties of folks in the lab, Those include biomechanics, physiology, prey-predator dynamics, and spatial ecology, to name a few. So room for a lot of different disciplines in this work. And so to motivate my pitch for physiological sensing, I want to walk you through what we know about this recipe for building the ocean giants that are the blue whales. So one of the keys to making an ocean giant is that, as you could guess, they need a lot of calories. And you can imagine a predator, whether terrestrial or marine, might do this in a variety of different ways. On one extreme, they could eat a ton of tiny things, and then at the other, they could eat fewer, larger prey items. In the case of baleen whales, which I'm going to get into a better description of in a second here, they've chosen the former, and they feed on highly abundant, really tiny prey. In the case of blue whales, they're krill specialists, so they're only feeding on krill. In the case of something like a humpback, they're more generalist, so they can feed on small fish as well. But they're feeding on these highly abundant tiny prey that are patchily distributed across the globe, generally in polar regions, but including up and down the length of the west coast here. Another key to being a giant ocean predator is that you need to have a reliable way to capture these prey. And blue whales have accomplished this by evolving a filter feeding foraging style called lunge feeding, where they capture giant volumes of prey-saturated water in their mouths and then filter the water out through baleen plates, which trap the prey in the mouth. Once all the water's filtered out, they can go ahead and swallow that prey. And this distinction, that of baleen whales versus teeth, like we might expect in a terrestrial predator, designates the two major groups of cetaceans. So for those who are less familiar with cetaceans, the two groups we think about are baleen whales or mysticetes, those that have baleen plates, and then the other group being toothed whales or odontocetes. I always remind myself orthodontist, those being the ones that have teeth. It's going to take a second, but I'm going to show you a video here of a blue whale that was taken by the American Museum of Natural History in collaboration with our lab. This is a blue whale surface lunge feeding. So blue whales sometimes do these lunge feeding events at the surface. Most often, they're doing them at depth. The nice thing about surface lunge feeding is that we can see it, and we can use UAS to actually capture it. And there are two things I want you to point out about this video, or that I want you to pick out about this video. The first being that you can actually go ahead and see that dense patch of prey that the animal's going after. It's coming at that prey at high speed, opening its mouth, and then closing it. And in closing it, you can see that there are several morphological features that are highlighted in this animal. So I'll walk you through these. The first being the ventral groove blubber. You'll hear us talk about that in our lab a lot as the VGB. The ventral groove blubber is a specialized type of blubber that looks like an accordion. And this blubber runs from the tip of the chin down to the belly button of these animals. And this is what expands when they go ahead and do a lunge feeding event. So they're opening their mouth, the accordion structure is expanding, and in the largest of blue whales, it's expanding to fit the volume of about your standard pool. So this is a huge amount of water that we're talking about here. You can also see the upper mandible, the lower mandible, of course, and then between the two inside of the mouth, you kind of get a sneak peek of what those baleen plates look like. And so the one final ingredient to this recipe for building an ocean giant is that there needs to be some reason that bigger is better. And I'll go ahead and caveat what I'm about to say here by saying that there are a lot of reasons why being bigger is better in the ocean. I'm sure some of you are thinking of some of those reasons in your head right now, but for the purpose of lunge feeding, this is one of those reasons, which is that bigger animals can engulf larger relative volumes of water on each lunge feeding event. And this is something that was revealed by Sherelle Kahane-Rapport, who was formerly a PhD student at Hopkins with Jeremy, now faculty at Old Dominion University in Virginia. And she took essentially all those metrics I showed you on the last slide and figured out a way to estimate the volume of each gulp. And what she did in the plot here from a JEB paper that came out in 2020 is she showed that if you plot total length of an animal against mass-specific engulfment capacity, so this is like how much are they gulping normalized by their size, this value increases with body size. So if you move from minke whales up to humpbacks, up to fin, up to blues, these bigger animals are packing more per punch, more per gulp in each of these lunge feeding events. And so this is really a morphological key for this recipe to life at the largest scales. And so when you combine in all of these things, the ecological ingredients, so that being the krill that are densely found patchily, but in dense patches, the biomechanical ingredients, that being the lunge feeding event itself, and the morphological ingredients, that being the ventral groove blubber and the baleen plates. It turns out that you can actually pretty robustly describe how baleen whales have gotten as big as they are. And this is an exercise that our lab went through back in 2019 prior to when I was in the lab. But they published this really neat paper that came out in Science. If you've not seen it, it's a really cool read, and I would suggest that you do. It's called Why Whales Are Big, Not Bigger. And this is the summary diagram from the paper, which essentially shows that return on investment in feeding increases with body size if you're a baleen whale, plotted in the reddish pink, but not if you're a toothed whale, plotted in blue. And so you can see here that basically all of these red lines have a positive slope, which say that blue whales are more efficient feeders. They're getting bigger return on investment every time they choose to feed. And that's why we have these really big whales in the baleen sort of grouping. And in the Odonocete group, we have this negative relationship, and this describes probably why we don't have so many big toothed whales. If you're curious as to why we don't have whales bigger than a blue whale, you should look at the paper. They describe it much more succinctly than I could. Okay, so to kind of get to the point that I've been building up to motivate, we have these ecological ingredients. There's so much work that describes krill distribution, patterns of diving on krill of different densities. We have the biomechanical component. We know what lunge feeding looks like. We know the speeds that are required to inflate that VGB and take these full gulps. We have the morphological ingredients. We know what the ventral groove blubber looks like and how it functions. We know how this scales with size. But the real key, and I'm sure you could guess from my title, is that this physiological ingredient is somewhat understudied. And I don't want to oversell this. There is some research in this domain. So there's one paper on blue whale heart rate that came out of our lab also in 2019, published in PNAS, that recorded the first ever heart rate of a blue whale. We'll talk more about that later. There's some publications that describe the respiratory behavior of blue whales when they're feeding, but really there's such a sparsity of information in this category that is baleen whale physiology compared to what we know about smaller whales and what we know about pinnipeds. And if I have not convinced you of that, there's this really cool quote that I think was published in a paper in 2009, that is that lunge feeding is the largest biomechanical event we observe on Earth. And we know so little about the how of how this extreme performance happens. And so I want you to recall this as I transition to the next slide. We know so little about this extreme performance in our biggest whales. And we know so darn much about the physiology of our best performing humans. So some of you might recognize this person. This is Kilian Jornet. He's a trail runner, a skier, and just generally has a ton of accolades in the discipline that is ultramarathons. And here you can see that he is conducting some sort of controlled physiological testing where he's running on a treadmill, and they're measuring all sorts of things with various sensors that he's wearing, some which are more invasive than others, for sure. But these are likely including things like heart rate, respiration rate, tidal volume, that being how big a breath is, inspired and expired gases, lactate levels, blood oxygen, body temperature, skin conductance, which is a proxy for the rate that he's sweating at, muscle contraction, and then omics. Omics being genomics, transcriptomics, proteomics, things that you might run from molecular samples you could take from blood, saliva, urine, things like that. And what's really interesting to me is that in this field that is human physiology, we know a lot about our best performers, but we also know a lot about not our best performers. I would put money behind that probably half of the people in this room, not that you all aren't physiologically amazing, but that many of you could call out your heart rate right now. You could call out your respiration rate. You could call out your blood oxygen saturation because you're wearing some kind of wearable that's measuring that in real time and telling you that at every minute of your life. And this is really neat, and I think we're at this exciting transformational time in these human wearables where we have access to a ton of physiological information that 5 years ago no one had access to. There's a website called Patently Apple, if you're really into this stuff, where you can track the patents that Apple files. And they've, in the last 6 months, filed several patents for blood pressure sensors in Apple Watches. So I would not be surprised if the next iteration of these watches can also tell you your blood pressure, which obviously has a lot of medical applications, which is really neat. And so compare this, how much we know about so many individuals' physiological performance, to the breadth of this. So we have cetaceans that are extremes in their physiological function in lots of different ways, most of which we know nothing about how physiology underpins this performance. So I've kind of harped on, and I won't continue about the largest animal on the planet, the blue whale. So this diverse cetacean taxa has not only our largest animal, but it has our deepest and longest diver. So the goose-beaked whale back in 2020 did a dive up to 3,000 meters. I actually think it was 8 meters short, but I'm giving it to him. And the dive was almost 4 hours long. And this is really crazy to imagine, right? So an animal diving to 3,000 meters and back is doing more than a 5K. You can imagine you doing a 5K, and they're doing this on a breath hold. It's really amazing mammalian physiological adaptation. Before I proceed, I do want to highlight, some of you in this room might be like, what the heck is a goose-beaked whale? I've never heard of that. And there's good reason for that. So in 2024, a group of colleagues that I know from Duke published this paper, first authored by an undergrad, which is really cool, calling for renaming Xiphias cabirostris, so that's a Cuvier's-beaked whale, to the goose-beaked whale because of sort of an exclusionary history associated with Cuvier himself. And so I would encourage you to check out the paper, but also just for your all's knowledge that going forward you might see Cuvier's-beaked whale referred to as goose-beaked whale. So in addition to the largest animals, the deepest divers, the longest divers, cetaceans also include the longest migrators in the gray whale. So these are animals that are moving nonstop for half the year. And then animals that are mammals that live the longest. So the bowhead whale holds the age record in the mammalian animal kingdom, which is pretty amazing. So taking all these things together, I'll go back to our blue whale and say that the point that I'm trying to drive home with this introductory section is that cetaceans are a diverse taxa. They're extreme performers in many ways, and we know so little about the physiology that supports this extreme performance, which motivates my interest in physiological sensing. So I believe that physiological sensing in whales is going to provide novel insights about the functional limits of life. And I think that's important from a basic physiological perspective and highlighting basic physiological principles, but there's also applications, as Gita mentioned in her introduction, to translational medicine. So how do these adaptations translate to how we think about cancer, how we think about aging, how we think about COVID or asthma? And then I think they also have applications, certainly in conservation. So how close these animals are to the so-called knife's edge, I think is important for how we think about conserving them. Okay, so I'm going to take a brief interlude to keep your brains engaged and give you a little information as to why the heck I have any ground to stand on to talk about biomedical devices and things like that. When I was a student going to seminars, my favorite part was always figuring out what the person's path was. I was like, how the heck do you get to a point in your career where you can stand in front of a room and talk about something for 45 minutes? I just want to know how these people are getting on this path. So I figured I would take a second and engage everyone's brains in a little bit different way and tell you about my career path. So I started actually in engineering. I went to Duke, born and raised in North Carolina, went to Duke for undergrad and did a degree in biomedical engineering, where, as you can imagine, we were doing a lot of learning about how do you build devices, how do you measure different physiological rates of interest, a lot of molecular scale stuff as well. Liked it, didn't love it. Did a study abroad semester at the Duke Marine Lab, which is pictured in that second picture there. The Duke Marine Lab is in Beaufort, North Carolina. Sort of similar relationship for you all with San Jose State, as well as Hopkins with main campus Stanford. And took a class in marine mammal biology taught by Andy Reid. He gave a lecture on diving physiology, and that was kind of my aha moment. Probably not in that moment, but some period of months after, I was like, oh, this is neat. I think like maybe my skills would be really applicable here. I can study a really cool system, bring unique skill sets that not everyone else has, and get the opportunity to be around really cool animals and be outside for part of my job. So that motivated me to pursue a PhD at Duke. So I stayed on, but did my PhD at Beaufort, so I lived there full-time for the duration of my PhD. The degree was in marine science and conservation, and for that I worked a lot with the smaller whales, so mostly Odontocetes, largely bottlenose dolphins, like the one you see here, and short-fin pilot whales, which we'll talk about a little bit later. And then I did both work with animals in human care, doing a lot of measurements, like sort of a lot of gold standard physiological measurements with animals in zoological facilities, and then also did some tagging work out off the coast of North Carolina, which I'll talk about later. And then I ended up here in Jeremy's lab, and I'm having a great time so far. Okay, so for the second chunk of the talk, I want to provide you all with 3 projects, some that are more complete than others, but basically 3 ongoing research projects that I'm involved in that highlight ways that I am and the teams that I'm working with are working to develop methods for physiological sensing in whales. So I'll go through these one by one, and they're really not meant to be exhaustive, so I would encourage if something in here piques your interest, certainly feel free to ask after or email me, but just sort of to give you a flavor of what these projects look like. And I'll start with one that Gita mentioned in my introduction, which is designing an electrocardiogram tag to measure heart rate in baleen whales. And I sort of bin this in the category that is hardware engineering. So how do we actually build tools that can perform for us in the field? Okay, so there are probably people in here who have heard this a million times, and I'm sorry if that is you. But for those who haven't, this is really the bread and butter of what we do in the Goldbogen lab. So we tag whales using devices that are produced by CATS, Customized Animal Tracking Solutions, which is a company based in Australia. And largely, these devices are suction cup attached, so they're non-invasive, and they contain depth and movement sensors. So we're measuring where the animal is in the water column, its orientation when it's there, and then also any fine-scale movements it might be doing. And then typically, these tags also have cameras on them, like you see here. One of the benefits of working with large animals is that you can get away with large tags, so we can actually fit full-on 4K cameras on these tags, which is really neat. And then the last thing that's on here is a radio for retrieval. So unlike maybe if some of you work with like acoustic telemetry tags or satellite link tags, this is fully archival. If we do not get this back, we get zip-zero data from it, so it's really critical we get it back. And the way that that works is after these tags stay on for anywhere from 6 to 24 hours, they're barely positively buoyant, so they will float to the surface. That antenna will stick out of the water, and then we use a radio tracker and antenna to actually literally play a game of hot and cold and go track them down, pick them up, bring them back to the lab, and download the data. Again, I know some of you have seen this, but for those who haven't, this is what the deployment looks like. So these are humpbacks in Monterey. You're seeing 2 views of the same tag deployment, and I'll let it run through a couple times. So you've got the tagger holding a long carbon fiber pole, the tag's at the end. When the animal comes up to take a breath, they just go ahead and pop that tag on. It's harder than it looks. That's a really heavy pole, and it definitely takes some training and finesse to do as good of a job as, I think this is Jeremy, as Jeremy does in this video. Okay, so to get to the ECG part, in 2019, Jeremy's group equipped one of these tags with an electrocardiogram. So electrocardiograms are sensors that use metal electrodes. If you've ever been to the doctor and gotten an ECG, you've gotten like stickers on you that have little metal touch points in them that measure the electrical potential as the heart depolarizes and polarizes. So the group, and which included Paul Panganis, who was Gita's grad school postdoc advisor, who's an amazing diving physiologist. They basically equipped this tag with the first ever ECG, put it on a blue whale, and it recorded this awesome 8-hour record where they got near continuous ECG data and from that could determine heart rate. And this is, even though this is an N of 1 paper, I can't undersell how much of a technological leap this was in our field. This is really amazing that they had this much success. And sort of to summarize, I guess the main take-home points are that these blue whales exhibit the typical mammalian dive response. So they have bradycardia when they're at depth, these low heart rates as low as 2 beats per minute, but typically in the range of 5 to 10. And then they exhibit tachycardia when they're at the surface in the range of 30 to 40 beats per minute. These are really low heart rates, but at their body size, this is generally what we would expect. And one of the cool extensions of this work that the authors do comment on in the paper is that if you look at the raw ECG data from this surface period, so each of these peaks, I'm sure there's some students in the room who are like, oh, I hate looking at this stuff. But each of these peaks is an individual heartbeat, and the way ECG works is that there's a very stereotypic waveform associated with mammalian ECG. And I'm going to go ahead and highlight 3 of these waveforms in color here. So the blue one, the pink one, and the green one. And the point of me doing that is to show you that these 3 waveforms are back to back to back. There's no space in between these beats here. And what that likely suggests is that for this individual, this is probably their maximum heart rate that they are physiologically capable of. There's no way they could squeeze any other beats into this, which is really cool. And so based on this work that, again, came out in 2019, the lab was, of course, interested in continuing it, but has had a lot of challenges in replicating this study. So that is what I am funded for as a postdoc, is to iterate on the tag design of this ECG tag and figure out how we can get it to work more reliably and consistently across different species, but even just deployment to deployment. And so here I'm just summarizing sort of the 3 main challenges that I've been addressing in the amount of time that I've been working on this project. The first being that we need to reduce the movement artifact, i.e. the tag jiggle. So these tags are not what I would call statically attached to the animal. They're attached with elastic silicone cups, which if you pull on them, they will give a little, right? And so when these tags are under high drag, when the animals are swimming quickly, they bounce up and down. We actually use that bounce to our advantage and can estimate speed from it, but that's a real problem in our ECG signal. So that's something we're working on. The other thing is we have to improve the contact quality of the electrode. So you can imagine if you went to the doctor and got an ECG and one of those stickers just falls off, it's not going to record well. So we need to ensure that the electrodes maintain contact against the skin regardless of what kind of sort of complex maneuvering an individual might be doing. And then we need to reduce flow around wiring an electrode. So anyone who's put anything in salt water, any kind of electronics, I should say, salt water is ionic. If you give it anything electronic, it'll short it if it can. And of course, in our situation, we're not going to get anything if these electrodes are shorted. And so that's a critical component, is that these electrodes make contact with the skin, but be totally isolated from salt water. That was sort of a mock-up of what the tag looks like. I think it's more clear in actually just seeing a picture of it. So this is what our current ECG tags look like. You see the typical CATS unit in the middle with 4 attachment cups. On the left and right, you have 2 electrode or suction cup embedded, electrode embedded suction cups. And that's a change from our earlier iteration. So we're sort of decoupling the sensing from the attachment here. And then one of the other sort of critical components that we've learned is that it really matters where these tags go. So there's a reason when you go to get an ECG done, they put those electrodes near your heart, right? That's where it's going to get the biggest signal. Well, in the blue whales, 1, they don't really show us their ventral side all that much. But 2, and this is a humpback whale, not a blue whale, but they have ventral groove blubber that runs down their entire ventral side. And so if you tried to stick a suction cup tag on that, it's not going to stick very well. And so what we've identified is on the left side this ideal sort of cardiac window whereby we think if we can put a tag there that's as close to the heart as we're ever going to get. And what I'm showing you on the right side of this screen are screenshots from CAT's video that actually recorded the tags in this exact position. So this is not something we're hoping for. This is a feasible position. It's plausible that these tags actually slid there into the drag shadow behind the pectoral fin, but the idea being that we can get tags to end up in this position. So I'll give you a sneak peek of some of the data that we've been getting from these newer tags. On the left, you see a picture of a fin whale wearing the tag. The data on the right is from a blue whale, to be totally clear. And what you're seeing here is data from a feeding dive and a non-feeding dive to similar depths. But what you can see on the left is that when the individual goes through these feeding events, so you see I've marked the lunge phase and the filter phase, that we see a spike in heart rate associated with that feeding event that we don't see when the animal's not feeding. And this is what we would expect and what we would expect based on the data we saw in 2019 as well. But we're getting this in a replicable way, which is really exciting. And it supports this idea of the high energetic cost of foraging. So this is where I'm going to end with this sort of project. But the point of it is to highlight that I think there's a lot of room to actually be working on building tools that integrate sensors that we know work in humans, but putting them in packages that can be deployed on these large whales. And that's something that I'm really excited about doing now and in the future. Okay, so to move on to sort of this second project that I'm going to talk about, this is changing gears from hardware engineering to what I would put in the bin of software engineering. And for this project, this is something I worked on at the end of my PhD, basically trying to figure out how can we use a bunch of data that we already have to determine a new physiological data stream from. So essentially, we put out these tags, and this project was on short fin pilot whales. We put out these tags and have been since the early 2000s. We have all this kinematic data, but we have zero physiology data to pair with it. But what if it were possible to pull physiological signals out of kinematic data? So that is what this project is focused on, and particularly thinking about breathing rate. So short fin pilot whales are something that maybe you all aren't familiar with. They're less common on the West Coast to my knowledge. They're more common out in Hawaii. But short fin pilot whales are highly social, almost always found in groups, deep divers that are found, I wouldn't say they're resident, but they are found very reliably off the shelf break in Cape Hatteras, North Carolina. So if you can see on this map, the last labeled thing on the left side at the bottom is Cape Lookout. That's the location of Duke's Marine Lab. And then this red patch essentially shows the highest kernel density estimate for the presence of these animals. Essentially, if you leave Beaufort and you go to the 800-meter isobath, you are pretty much guaranteed to see pilot whales. And this is a plot of a bunch of dives recorded by D tags, which are a similar type of tag to the CATS tags, just to support this point that pilot whales are deep divers. So they're not as deep as some of the species that we all study, but regularly performing dives to 800, 900, 1,000 meters. And what motivated me to be interested in these animals is that previous work had suggested that the aerobic dive limit, the calculated aerobic dive limit for these individuals was around 14 minutes. And they quite regularly are exceeding that dive limit. And I would say the general sort of discussion around pilot whales at Duke was just that they're really high-performing divers for their body size. And so this was sort of intriguing to me from a physiology background. If pilot whales are at all interesting to you, a paper came out that I'm really happy about because it was co-first led by me and an undergrad named Lauren Miller, who graduated from Duke a couple years ago, and she's now at Duke Med School. And so we worked on this together for her senior thesis, showing that social association and local bathymetry probably drive the diving behavior of these animals. And this is one really neat plot that I wanted to share, which shows individual distributions of dive durations of all the individuals included in the study. And the colored outlines represent individuals who were tagged together. And in most cases, these were not individuals of the same body size, the exact same body size at least. And what you can see is that they're really similar qualitatively, and we have some quantitative support for this too. But they're exhibiting really similar dive patterns with these individuals that we believe they're associating with. Okay, so to get back to the breath durations, this is what it looks like when you go out to the 800-meter isobath off of Cape Hatteras. This is what we call pilot whale soup. They are everywhere, and they do this behavior that is called logging at times where they sit at the surface, and it looks a little something like this. And the reason that I'm showing you this video is that logging makes detecting breaths kind of hard. And the reason for that is that if you were trying to do breath detections for a blue whale, generally, a blue whale is going to come to the surface, take a breath, go down to 5 or 10 meters for a little, come back up, take another breath, and repeat, rinse and repeat over and over. So it's pretty easy to pick out breaths. Because pilot whales do this logging behavior, the whole time of which they're certainly breathing, it's not that trivial to pick out individual breaths from a depth record alone. And so as I was in the PhD thinking about, is there a way in which I could do this, I thought, well, these D tags are deployed often close to or on the thoracic cavity. They're recording movement data. Perhaps every time the animal takes a breath, there's some amount of movement that's happening that's being recorded by these tags that we could use to extract breathing events. So I'll drop in these arrows here. If you squint, maybe you can see them. But what I noticed is that during these periods of logging, there were pretty consistently these sort of little buzzes. They looked like high-frequency kind of jiggles fairly regularly interspersed throughout logging periods at the surface. And I suspected these might be exactly what I just described. These might be movement associated with breathing. The nice thing about D tags is that in addition to motion, they also record acoustics. So D tags and cat tags are kind of like cousins. The cat tags are good at video. The D tags are good at acoustics. And so we have this acoustic information, which allowed me to go ahead and just listen. So say I saw 6 of these little squiggles. I could listen in that same time interval and go ahead and audit it and say, hey, do I actually hear 6 breaths during that period? That's happening at the same exact times that I'm seeing these squiggles. And I'll just play a little bit of this for you to show you just how clear these breaths are. So I'm not going to play the whole thing. It's only about a minute, but it contains 6 breaths, which are associated with those 6 accelerometer sort of squiggles that you see in that middle panel. So there you go. That's what a pilot whale breath sounds like. Yeah, so they're pretty distinct. They're pretty easy to pick out, particularly if the tag is placed right behind the blowhole, which many of ours are strategically. So this was exciting. So this said to me, in fact, these squiggles are breaths, and if I can detect those, I can get a continuous breathing rate from kinematic data alone. And so as a chapter in my dissertation, I wrote this MATLAB tool package that I called RESP-Detect. I kind of regret it because it's a little bit of a tongue twister, but I published it, so I'm stuck. But RESP-Detect is meant to take a tag file, so for us those are PRH files, pitch roll heading, and automatically detect breathing events and basically publish those to you in some kind of map file that's easy to work with for a variety of analyses that you might want to do. So now you have your dive profiles, you have your feeding events, you have your calling behavior, and you can add to that breathing rate. And this is all publicly available on GitHub, and it's currently under review in JOS. This is all fine to share. JOS is totally open. You can see my active reviews if you want to log in and look at them. But JOS is a really cool journal if you've not heard of it. You can publish these code packages, and you get a DOI for them, but also it makes it public and very reproducible for other folks to use. So RESP-Detect exists online, and my goal of that is that maybe other people would use this to add a physiological signal to their whale tag data sets. So in the interest of time, I'm not going to go too far into the weeds of how this works, but I do kind of want to show you what a few of the steps look like to sort of convince you that it does work, I guess. So here we're looking at one record of a short fin pilot whale tagged in 2008. The tag stayed on for 3 hours. The animal did 36 dives that were greater than 20 meters deep. And what you see in the middle there are 3 panels that represent 3 different movement parameters that have been processed in a variety of ways. And they only exist for when the animal was logging. So I go ahead and figure out when the animal's logging. I only care about this stuff when the animal's logging. And then the asterisks designate all the local maxima of peaks in each of these different segments. And then essentially what you're seeing on the right is highlighted in pink sections of the dive profile where 2 peaks across these three signals co-occur within some small window of time that I define. And I'll zoom in to make that a little more clear here. So again, where two of the three of these signals have local maxima, co-occurrence of local maxima, on the right, highlighted in pink, is a probable breath event. And this is some window of time that I'm saying, okay, it's probably likely a breath occurred here because we're seeing this spike in movement. And then it goes ahead and pretty simply defines the midpoint of each of those periods as when the breath occurred during this logging period. And so this might seem trivial, but there's a lot of signal processing that goes behind this, and ultimately the utility is that you can take a tag record and spit out every single breath the animal took. The pink ones are the logging breaths, so those are the ones that are influenced by the movement information. The blue ones are those single surface, or what I called, I think I called them like single breath surfacing events, and those are the ones more similar to blue whales, where they come up, are there for a short period of time, bounce down to 5 or 10 meters, and rinse and repeat that. And so if we zoom in, that's what that looks like in a little more detail. So that's every single breath event marked in time across the dive profile and these three movement data streams. And then from this, you can ultimately derive continuous breathing rate. And this is like, the actual point, which is that you can now look at how these animals are modulating their breathing rate relative to a dive, for example, or say they're exposed to something like sonar and you want to know what their response is to that. This might be a useful signal to look at. So, take-home point here being that not only can we build new things that can measure physiology, but we can extract physiology from data we already have, which I think is really cool, and that's why I'm excited about this project. Okay, so getting to the last project that I want to share with you all. I think we should be good on time here. And that is a project looking at molecular methods for studying cetacean adaptations, particularly to hypoxia, which is the low oxygen conditions that are created during diving. I'll tell you why we cared about that for this specific project in a second. But I think about this as a systems engineering project. So this is thinking about, we have all of these sort of emerging technologies in molecular biology that we apply to humans, that we apply to mice, that we apply to a lot of traditional model organisms. And for the most part, no one's applying these to whales. And so I think there's huge leaps to be made in this arena, is how can we translate sort of these systems of investigation we already have to a non-traditional model organism. Alrighty, so the motivation for me even starting to think about molecular work was that I met Dr. Jason Somarelli and Dr. Will Eward, who are part of the Duke Cancer Institute, and we worked with some other folks at Duke, and we're talking a lot about adaptations to hypoxia because they are both cancer biologists, and in cancer tumors, one of the main drivers of metastasis is low oxygen, and what they were both calling upon was the fact that marine mammal tissues, in theory, go hypoxic all the time, and dolphins and whales' cells are not moving in response to hypoxia. So somehow, these animals have disentangled hypoxia from motility. So they were really interested in that. We started working together. We published this paper, which was a study that I'll tell you a little bit more about in a second. This is one of the chapters of my dissertation, but I would call out to the students that I think like working across these interdisciplinary boundaries is really ripe in our field. You send someone an email and you attach a picture of a dolphin to it, they are a lot more likely to respond, and that is essentially what resulted in a lot of these really cool collaborations. So a little bit of motivation, I won't bore you with this, I know you know this by now, but if you read most diving physiology papers, you'll see somewhere in there some version of the term dually constrained. So marine mammals are dually constrained. There's two things they need to live. One is air, that's only at the surface. One is food, that's only at depth. And so they have to perform breath hold dives to access these underwater resources. And we think about adaptations at scale, so whole organism things that might be like diving within your aerobic dive limit, organ and tissue specific adaptations that might be sort of the marine mammal dive response and responses to apnea. And then there's also this level of cellular and molecular adaptations, which again, is I think, newer because the technology is new, but I think it's understudied and in a really exciting area. So when I got together with Jason and Will, I had been working at Dolphin Quest Oahu, doing some heart rate measurements and respirometry work with dolphins. And we chatted to the folks there and we're like, hey, could we take molecular samples like blood or urine or something when the dolphins are doing breath holds and see if there's anything we can see? Is there any sort of transcriptomic response that happens when these animals do breath holds? And bottlenose dolphins are not the best breath holders in the world. That is not beyond us. But they are really well trained at zoological facilities and allow us to do these kind of experiments. So they said yes. So we did experiments where we had bottlenose dolphins doing breath holds. We pulled blood samples at the start, middle, and end of the breath holds. As we expected, hypoxia was developing across with time. And we found a couple of things. We found 2 genes that were significantly upregulated, and this is all RNA-seq data, so this is all at the level of the transcriptome, not at the protein level, which some might say is really what's making the difference, but sort of in the path, demonstrating upregulation of some important genes. These 2 genes might not mean anything to you. They didn't to me at the time, but as we dug into the literature, we learned some things about them. So we learned that ALOX5 is a gene that controls the regulation of a small molecule called lipoxygenases. And these molecules in humans and other traditional model organisms regulate and induce vasoconstriction. And for those of you who know diving physiology, you know vasoconstriction is a big part of the dive response. And this is pretty exciting because it suggested to us, okay, maybe there are some things going on that we actually can measure at the molecular level that are supporting these adaptations to diving. Another really exciting thing is that IL-5RA, which is this other gene that we saw was significantly upregulated, is associated with asthma and actually has been targeted by an FDA-approved drug for the treatment of a severe type of asthma. And if you think about marine mammals experiencing low oxygen conditions, asthma is a condition where low oxygen conditions in the tissues are created. There's certainly some crosstalk there. And so this was really cool to us because it suggested, in this case it wasn't the case, but maybe in the future, these sort of studies can identify molecular targets of interest that could be targeted for therapeutics for different relevant diseases. Okay, so probably many of you are thinking, well, that's neat, but you're not going to get a goose speak well in a zoo or aquarium. So how are you going to study this in the most extreme species? And the answer to that is that between Jason, Dr. Jillian Wissey, who is one of my lab mates in the PhD, and myself, and many others in the lab, we worked to develop a process to start tissue cultures from cetaceans. And so we take blubber skin biopsies from cetaceans all the time. It's what we use to get genetic sex of individuals. We don't need the whole sample for genetic sex, so we often have extra. And we can take some of that sample, cut it up in a somewhat specific way, plate it on a tissue culture plate with nutrient-rich media, and grow cells, and in this case, fibroblasts, from these tissue explants. And this is really cool because it gives us a model to be able to do controlled experiments in the lab with an organism that we're never really going to be able to control. And just to wrap up, I'm going to give you a sneak peek of a couple experiments that we've done with these cells to demonstrate what I think the power of a cell culture model is. So we can take cells, we can expose them to normoxia, that's just normal conditions. We can expose them to hypoxia, so grow them in low oxygen conditions, take the RNA out of them, run RNA-seq, and look at transcriptomically what's happening. And one of the things that we looked at first is the regulation of genes in the glycolysis pathway. So this is a heat map. Each row is a gene in the glycolysis pathway. Each column is a species. And the color of each cell represents whether in hypoxia the expression of that gene went up, which is red, or down, which is blue. And so in humans, when they experience hypoxia, they turn on glycolysis. Glycolysis is a form of anaerobic respiration. This is exactly what we think they should be doing. But when you look at some of our cetaceans, particularly the deepest divers, so the sperm whale and that should be updated, goose-beaked whale, we see that they're not just blindly turning on all these genes, and in fact, some of them are actually being downregulated, which suggests to us that somehow cetaceans have uncoupled hypoxia from glycolysis in a way that humans have not, and they're exhibiting this sort of really interesting tempered glycolytic response. So this is something that folks still at Duke have been working on more since I left. And the last thing I'll leave you with is that you can actually just even look at the cells themselves. So this is a human fibroblast on the left, a goose-beaked whale fibroblast on the right. The nucleus is stained blue, the actin's in green, and the mitochondria is red. And it's pretty qualitatively obvious that the goose-beaked whale has a lot more mitochondria in the cell than the human. And this is true across cells, including when you control for cell size. And so this is something that, to us, is quite interesting because high concentrations of mitochondria might help these animals take advantage of low oxygen conditions, but they're not energetically cheap to keep up with. So we don't really know what the whole story is here. But I'm going to leave this project here and summarize it by saying, I think from the systems engineering perspective, the idea that other fields might have systems of investigation already developed that we can pull some of these extreme species into as a non-traditional model organism, I think is really powerful and something that I'm excited about continuing and am continuing in a couple of different ways at Hopkins. Alrighty. So in summary, I've showed you some hardware methods we're using for physiological sensing. I've showed you how we can engineer data that we already have to sense physiology. And I've showed you that I think there are some really exciting molecular methods that are going to tell us about physiological function that are sort of standard in other fields and that can potentially be quite easily translated to our fields. So in sum, we've got the ecological ingredients, we've got the biomechanical ingredients and the morphological ingredients, and I think this is a really exciting time to be working to fill the gap that is these physiological ingredients and translating some of these physiological sensing methods that are really common for the everyday human, but also the extreme performer in the human world to the extreme performers of the animal kingdom. And with that, thanks for your attention, and I'm happy to take any questions.
I can start off. So I've also been trying to develop the heart rate tag. So have you been able to get continuous records, or are you still getting gaps?
It's still quite gappy. And one of the interesting observations that's ends in the single digits, but is that we get more continuous data right when the tag goes on, and then as the tag stays on, it goes away. But then we'll have some periods, like hours, but after hours of having not one single detectable beat, where it comes back. And that I can't quite wrap my brain around what would cause that. So the answer is not continuous. I don't know. I don't know what the key is, but I think that's a challenge. And I think one way is you just keep building up these chunks at different time points and paint the story that way. But
I'm working on a paper right now where I did that. Basically, to get one dive thing, it's 3 whales.
Yeah.
So
yeah.
I was like, are you
It's
a
hard project for sure.
Any other questions? This is a good warm-up for some of you in class tomorrow. You are going to be looking at heart rate signals, and you will need them by the end of class.
Do you have an idea of how we could get the CATS tags behind the left pec?
Yeah. That's a great question. So one of the things that we have been thinking about, I think the pie in the sky, which has come up in meetings that I've been in, is like a tag that knows where it is on the body and could actively move itself. That requires a lot of onboard processing that we just do not do. Our tags do not do that in any sense. So I think in the absence of that, the way that we've been thinking about it is, can we build, say we know the tag's going to go on the left side, so we have some idea of what the flow is going to look like given it's on the left side, could we add like a wing or a spoiler that encourages it to slide down? So maybe different than some of the like penguin tags that are getting, you know, glued on or something, or like a seal tag that's getting statically put in place. These tags do slide. And so that's one of the things we've been thinking about is like, can we put it on the top of the animal, but have something that encourages it to slide down into that drag shadow? But if you are engineeringly inclined and have ideas, we're open to them.
This is just another thought to add on to that, because you've obviously done work with captive animals as well as wild animals. So, I don't know, this is a silly, I don't know, but have you reached out to them to see if they would be open to testing the tags on their animals in a smaller, more controlled setting to look at different designs like that?
It's a great question, and we have. So we have worked with SeaWorld San Diego and tried these tags there. There's a couple challenges in trying to take a tag that's meant for a blue whale and put it on a killer whale. One of them being that the killer whale, like the curvature of the killer whale surface is more. So our tag is pretty wide. You know, it's like yay wide. You can pretty much assume that across that distance, the blue whale is like a table. Like it's perfectly flat, and that's how we build our tags, is assuming they're a table. The killer whales are not a table at that, you know, that distance. So our tags don't stick super well on them. When we've tested them, just the width of them, like the electrodes don't make great contact. The other challenge is that we have tested the tags on stationary killer whales, and they work great, and it's all very clear, including when it's up on the lateral surface in a position that would be more taggable. When they start swimming at high speed, because they're like turning, like the pools are not so big that when they're turning, they're doing pretty high acceleration turns, and the tags tend to slide off like immediately. Yeah.
To add on to that, what about testing a non-functional, like third-scale model that is more appropriate to build a size that aspect of it?
Yeah. Yeah.
If we're talking specifically about moving the tag down or getting
Right.
to move into that drag shadow,
Yeah.
then that's just sorry.
Yeah, no, that's a great. It's a great idea for that idea of like, how are we going to get a tag to move? We are, in terms of like the functional tags, they are about as small. It's shockingly, they are about as small as they can get.
Yeah.
But like, we could totally have a dummy tag.
But more so for teasing out
Right.
the shape aspect
Yeah.
of getting that shape right, but you then scale up
Right.
to
Yeah.
something that actually can accommodate all the sensors.
Yeah, I love that. That's a great idea. I hadn't thought of that before, but that's a good idea. Yeah. Cool. Wow. Yeah. Come on down. Yeah, thank you guys.
She's going to be back in a few weeks for class, so if you think of anything.
Yeah. And I'm like 20 minutes down the road and not bad traffic, so.
So yeah, thank you very
Cool.
much.
Thanks, everyone. Hey, thanks so much. Yeah. Yeah, it's cool stuff. We just got a bioX grant, which is Stanford's like biology, molecular, whatever, like